Welcome to Translating Proteomics, where we explore the science of proteomics and its growing impact on basic biological research, drug development, and more. In this episode, we cover the central dogma of biology and specifically discuss what the central dogma actually says, why it's important to develop biological models that account for regulation, and how new tools will help us make better models of biology. And now your hosts, Parag, Malik, and Andreas Humer of Nautilus Biotechnology.
Thanks so much for joining us today. Today we're going to talk about the central dogma of biology and where it goes wrong and how it's terribly misunderstood. Just to give us some background. Many people, when they think about the central dogma, they think that it implies how all of biology is regulated, from DNA to RNA to protein. And that what happens at the genome level happens at the RNA level, happens at the protein level. So from high school biology on up, we're taught that information in biological systems flows from DNA to RNA to protein. And this concept has permeated all of biology. And yet, I would argue that this is a gross misunderstanding of the central dogma. And so we're going to talk about it today and how it applies to proteomics, how it applies to protein regulation, and beyond. So I'd like to start off, Andreas, by just asking you, point blank, have you read the 1958 paper by Francis Crick on the central dogma?
Absolutely not. But I've certainly seen, references to, it through diagrams and images where the arrow goes from the DNA on the left side to the protein or metabolite on the right side. And, literally this was a, logical diagram, or is presented as a logical diagram in a way that obviously is not true.
So coming back to the central dogma, I think we do have that picture in mind. We have that picture of an arrow going from DNA to RNA to protein, and that picture is in every single biologist's head. But when you go back, as it turns out, that concept of the central dogma was first presented by Francis Crick in a lecture in 1957 as part of the Society for Experimental Biology Symposium. And really, what he was talking about was, where do proteins come from? When we go back and we look at that paper, he was not saying anything about how all of biology is regulated. He wasn't even saying about how cells work. Really, at that point in time, when we go back, in 1957, ribosomes were only known as microsomes. Their function wasn't known. We hadn't even really discovered messenger RNA. And yet, somehow, what he was trying to ask the question, where do proteins come from?
First of all, I personally think about, when I see the central dogma of biology, more like this is how they biochemically relate, rather than how they are controlled.
Yes.
However, if you think about the last 30, 40 years in life sciences, the fact that we had this revolution of genomic tools that obviously dominated the conversation about how genes are the critical piece in a lot of biology.
Yeah.
Despite the fact that in many cases that's not true. We know it's true, for example, for genetic diseases, right? Yeah. A particular gene has a defect, a mutation, like in cystic fibrosis, it will have an effect on how the protein is regulated or not, or how the protein is functional or not. So I think those examples have become sort of the main story instead of the side story. And I think we have a lot of work to do to reverse that trend of people thinking about the gene being the all, determining factor in organisms.
Yeah, so I guess for me, oftentimes what I think about, we care about if I perturb a system, if I change it in some way, if I knock out a gene, if I mutate a gene, if I am exposed to sunlight, all of these different variations, these perturbations have a consequence. They have a consequence in you being healthy, you being sick, you being alive. And so what we want to be able to do is predict those consequences. We want to understand the system well enough that we can predict those consequences. And when we look at the flow of biological information, yes, biochemically it is true. we go from DNA to RNA to protein. Mechanically, biochemically, we get proteins starting from a blueprint of DNA. But that doesn't help us predict what might happen, how a perturbation might impact a biological system.
And I think the perspective you describe is so predominant and, in general, is because we have a mechanistic understanding of how biological systems work. We literally look at them as mechanical systems, and we want to apply the laws of physics to them to explain them. When it is not correct, and as you described correctly, biological systems are built to dynamically adapt to the environmental changes, and they have to be built in a way that are not predictable, because nobody can predict what's going to happen 1,000 years to humans from an environmental perspective. Nobody can predict what happens to microbes or plants 500 million years from now. Nobody knows. And so biological systems are not deterministic systems. And I think that is the key message that we need to sort of keep communicating to others, that, the central dogma of biology may be useful in describing how they're biochemically related, but they're not useful in explaining biology.
Yeah, and I think that's right. I think that's the key point of the misunderstanding of the central dogma, is that it is not an explanatory tool about regulation or dysregulation. It is a very functional explanation of where proteins come from. And it's led to, oftentimes I see these misunderstandings. I see people saying, well, RNA and protein should perfectly correlate with each other because protein comes from RNA. We have so many examples where you measure both RNA and protein and you plot their relationship to each other, and they're completely uncorrelated. And so to me, this is very natural. But I'm curious, from your perspective, going back in time, going back to when we first started being able to measure in mass proteins, this observation of the discordance between transcriptome and proteome, where did you first see that?
I think it emerged with really the emergence of better or more mRNA tools. And I think because these tools turned out to be really useful in areas like developmental biology, where in fact the translation of RNA directly correlates to proteins because you synthesize new proteins. Mm-hmm. That's when everybody sort of conceptualized or took that concept and then made a rule out of it when it actually is not a rule, right?
Yes.
I think there's probably misconceptions that sort of made it into general biology book that should not have been generalized in the first place.
Yeah, and so when we think about regulation, when we think about going from DNA to RNA to protein, there are a number of steps in there where there's regulation happening. There's the rate that RNA is transcribed and degraded. There's the rate that protein is translated and degraded. And there are these levers that cells are using constantly to make choices and to sense and to respond. And so under the hood of the central dogma, beyond the central dogma, are those levers. And I'm curious, from your perspective, what are the levers you believe are most impactful?
Well, let me look at this very differently.
Yeah.
I think now we already understand from the biology we have discovered, including genomic and proteomic tools, that there's many levels of control and feedback loops. And it's truly more about the feedback loops rather than the one directional flow of information. So, for example, we know that histones control the access of transcription factors to particular genes, right? Mm-hmm. And in fact, those histones themselves are controlled by other proteins, which we appropriately name writers and or readers and writers. Mm-hmm. And in these cases, the histone tail or the histone protein is modified to the point where it wraps the DNA in a very different way and makes it either accessible or inaccessible. So this is a mechanism that allows the protein or the proteome to control what actually happens on the genomic level. And so I think we already have plenty of examples where we see information flow in two directions.
So, but what you're describing, this temporal, cyclic, dynamic, sounds complicated. And just from a practical perspective, can we ever make sense of this? Is this a tractable problem to understand how biology works?
Maybe we don't have to understand every single aspect of control. I think as long as we respect the fact that biological systems are, dynamic systems that are, built to adapt to environmental challenges. And I think then in that case, we'll have a much better way of really explaining the findings we have in science, right? The counter argument to all of this is that I think we need many, 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 many more data points when we, look at all these dynamic aspects of biological systems. Mm-hmm. I think we just don't have enough data points today to really be, escaping the central dogma of biology, right? It's such a simple concept. It's so simple to prove. It's so simple to observe. I think if we get tools where all these data points provide us with more information, I think we'll, let go of that very quickly.
So let me argue with you just a little bit. I think, so I agree with you. We don't necessarily need to understand every little detail in order for it to be useful. On the other hand, the more there's a famous quote attributed to Einstein, or maybe Feynman, that which I can't create, I don't understand. And that's the root of synthetic and systems biology, is saying, let's understand all these pieces so we can put them back together again and really understand them. So I think the quest for understanding regulation through all of those levels of detail is critical. And what we're talking about are models. The central dogma is a model that is used ubiquitously throughout biology. But it sounds like what we need is a new model that describes not just how biomolecules get created from each other, but how cells work.
When I talk to, scientists I work with, I think that this particular insight that we have a very simplistic perspective of biology today is starting to change. Even scientists that are focused mostly on genomics are now approaching us in the proteomics field and saying, we need this protein level information. We know that there's a lot of changes happening. And I think what's missing is really the model as you describe. And in fact, I would go even one step further. I think we don't have a general theory of biology. If we had something that the physicists have, which have a general theory of physics, we would be able to probably design models and build models that could explain biology in a much better way. But in the absence of it, that's something to be worked out.
So just putting you on the spot, I'd love to hear an anecdote from chatting with somebody in the field or outside of the field, and having this conversation about, I only need to measure the RNA. I don't need to measure the protein. And how you had that conversation, how it went, and how you argued, and if you were successful.
So I think the excuse that a lot of people brought up, even if they knew somehow that maybe mRNA wasn't really representative, was the excuse of it's just easier to do, right? The proteomics is so complex, it's so involved, it takes so much time, I need so much sample, I can't amplify my signal. But I think that's going to change. Mm-hmm. And I think if we have tools that allow total access to the proteome, at all levels of sensitivity, massive quantities of data points can be accumulated, I think that perspective will probably change, because that was the main argument, not to look at the protein.
So it wasn't that proteins weren't important.
Yeah.
It was that, hey, we can't do it.
Yeah, exactly. It was more about we can't do it rather than we don't accept the role of a protein in that sense, right?
That's interesting. So you didn't hear, hey, I've got the transcript, that's good enough. You heard, I would like to get to the protein, I just can't.
Yeah.
So again, going back in time, there was a world where we didn't know what the ribosome was. There was a world where we didn't understand where proteins came from. Over time, we started to understand these links. Think forwards for a minute. Again, we're 10 years from now, 50 years from now. How have the tools that are emerging further changed our understanding of models that are appropriate for biology.
I have been talking a lot about information density and number of data points that you need to understand the the dynamic nature of the relationship between those different levels of biological control. I think the other part is really getting the sense, getting the resolution, right? I think a lot of biology happens not just by transcription, but it happens by changes in individual proteins. And so I think that requires, again, tools that are differently constructed than the tools we use today. I mean, a Western blot simply cannot capture the dynamic of a protein. It's just not possible. And so it basically replicates the static picture of an RNA measurement. Mm-hmm. And so I think the tools moving forward, if they can capture the dynamic nature and the intricate details of changes we expect are happening in biological systems, then I think, in 10, 15 years, we'll have a very different perspective of how biology is controlled. And maybe the central dogma is going to become, a part of history.
We've had a great conversation today about the central dogma and about how misunderstandings of what it was trying to say about how biology works have permeated throughout the field. And really that the central dogma describes the biochemical relationship between biomolecules and not the regulatory relationships under the hood of how cells work. We've talked also about how regulation is more complex, but also more difficult to grasp and more difficult to study. And then we also discussed how there's a tremendous need for new technologies that are comprehensively able to measure all of these different molecules across space and time to really develop better models for how biology works. Thanks so much for joining us. Looking forward to seeing you again next time.
We hope you enjoyed the Translating Proteomics podcast, brought to you by Nautilus Biotechnology. To contact us or for further information, please email translatingproteomics@nautilus.bio or scan the QR code on your screen.